Hi everyone, in this presentation we're going to be focusing on the major accessory organs of the digestive system, which are the liver, the gallbladder, and the pancreas. So let's get started. We will start with the liver, which is the largest visceral organ, and it is one of the most versatile organs in the body. Most of its mass, as we can see from this image, will lie within the right side of the body in the upper right quadrant. The liver will weigh about 1.5 kilograms, which is about 3.3 pounds, so it's pretty heavy. And this large, very firm, and it's sort of reddish brown organ will provide essential metabolic and synthetic services that fall into three basic categories. And these are metabolic regulation, hematological regulation, and bioproduction. And before we talk about these essential metabolic and synthetic functions, let's talk about the anatomy first, as I think it will make it easier to understand the function. Now, today, there are more than 200 different functions that have been attributed to the liver. And here's a partial listing of these functions. Therefore, as you can imagine, any condition that severely damages the liver will represent a very serious threat to life. The liver does have a limited ability to regenerate after injury, but liver function will not fully recover unless the normal vascular pattern returns. With regards to the anatomy, on the anterior surface, we find this ventral mesentery that's called the falciform ligament, which will mark the division between the left lobe and the right lobe of the liver. At the end of the falciform ligament, there is this thickening that's called the round ligament or the ligamentum teres, which is this fibrous band that will mark the path of a degenerated fetal umbilical vein, meaning that during embryonic development, the umbilical vessels carried fetal blood back and forth to the placenta, with the umbilical vein carrying oxygenated blood with nutrients from the placenta to the fetus, and the umbilical arteries transporting deoxygenated blood with waste products from the fetus to the placenta. And after birth, when the umbilical cord is cut, this is what is left over, the ligamentum teres, or round ligament. Now, the liver is going to be suspended from the inferior surface of the diaphragm by what we call the coronary ligament, which is this one right over here. Therefore, we can only see the coronary ligament on a superior view. The shape of the liver will conform to its surroundings. Therefore, we can see that the anterior surface or the parietal surface will follow the smooth curve of the body wall. However, the inferior surface or the visceral surface will bear all the impressions of the stomach, the small intestine, the right kidney, and the large intestine. Now, the liver has classically been described as having 4 lobes. We talked already about the right and the left lobes. Now, the impression left by the inferior vena cava, which is this one over here, will mark the division between the right lobe and the small caudate lobe. Inferior to the caudate lobe will lie the quadrate lobe, which is going to be sandwiched between the left lobe and the gallbladder. So again, this one here is the caudate lobe, and this one over here is the quadrate lobe. The quadrate lobe on the model, it looks a little bit more squared. So that's how I'm able to remember that this is the quadrate lobe. Each lobe of the liver is going to be divided by a connective tissue into approximately 100,000 liver lobules, which is going to be the basic functional unit of the liver. A typical liver lobule is going to be hexagonal in a cross section, meaning that it has 6 sides. So let's count. 1, 2, 3, 4, 5, 6. And in each of the 6 corners of the lobule, we will find 6 portal areas, or also called hepatic triads, or even portal triads, like the image shows. And what will these portal areas contain? Well, they will all contain 3 structures. That's why they're called triads. They will contain a branch of the hepatic portal vein, a branch of the hepatic artery proper, and a small branch of the bile duct. And where are these branches coming from? Let's start with the branch from the hepatic artery proper. Remember that the first branch off of the abdominal aorta was which one? The celiac trunk, right? And because it is a trunk, it means that it is bigger than an artery, therefore it will probably split. And in fact, it does. It splits into 3 branches. Do you remember the 3 branches? The one that goes to the left is the splenic, which, like the name says, it's going to the spleen. The second one will move up towards the left, which is the left gastric. And the third one will branch to the right and become the common hepatic artery. On this model, we can see the celiac trunk with the branch going to the spleen, so the splenic artery. This one that is sticking up is the left gastric artery that will supply blood to the stomach. And this one going to the right side is the common hepatic artery. It might be a little bit difficult to visualize this, but the common hepatic artery will split right over here into 2. The one that comes down to the duodenum and also to the stomach, which is not represented on this model, is called the gastroduodenal artery. And the one that goes into the liver is called the hepatic artery proper. So this one is the hepatic artery proper. So is this one here. And so is this one here going into the liver. Once it enters the liver, it will split. So all of these arteries colored in red, since they are a branch off of the original abdominal aorta, therefore they are carrying oxygen-rich blood, they will represent a branch off of the hepatic artery proper. And we can see this branch at each corner of the hexagon as well. Now let's talk about the branch from the hepatic portal vein. Remember that all the blood from the digestive system will exit through veins that will merge to form what we call the hepatic portal vein. Therefore, on these models, we can see the splenic vein coming from the spleen, the inferior mesenteric vein coming from the left side of the large intestine, therefore mainly from the descending colon. And over here we see the superior mesenteric that will bring blood from the opposite side of the large intestine, therefore mainly from the ascending colon. Of course, there are veins from the stomach that will join as well. And they all come together to form the hepatic portal vein that will enter the liver. And we can see it right over here on this image entering the liver. Now, why does this happen? Why don't these veins just merge onto the inferior vena cava and take this blood filled with waste products directly to the heart to be replaced by nutrient and oxygen-rich blood in the pulmonary circuit? Well, one of the functions of the liver is to filter this blood, and this is extremely important. And why? Well, imagine if you eat something that is toxic to your body, and imagine if your body did not have the liver to filter this blood. Then this would mean that the blood will go directly into the inferior vena cava. From there, it will go to your heart, then to your lungs. From your lungs, where would it go? Well, it will go everywhere in your body, through the systemic circuit, including your brain. And these toxins could actually kill you. So this is the importance of the portal system and one of the very important functions of the liver. Now, this filtration is done by the cells in the liver, which are called hepatocytes. So again, here's the hepatic portal vein that once it enters the liver, it will split into branches, as we can see right over here. And these branches will be present in each of the corners of the hexagon, together with a branch of the hepatic artery proper. And the third structure that is present in the triad, because it is called a triad, right? So this means that there are three structures. And this third one is the bile duct. Now the bile duct will do exactly this. It will transport bile. Which bile? Well, the bile that is produced in the liver by the hepatocytes. There we go, another function of the hepatocytes. And we will talk more about the function of bile later on. Going into the lobules, we can see the formation of these structures that are called sinusoids. And branches from the arteries and veins, they will deliver blood to the sinusoids of adjacent lobules. And as the blood flows through the sinusoids, the hepatocytes will absorb or secrete materials into the bloodstream across their exposed surfaces. Now, the fenestrated walls of the sinusoids contain large openings that will allow substances to pass out of the circulation and into the spaces surrounding the hepatocytes. Then the blood will leave the sinusoids and enter the central vein of the lobule, which, like the name suggests, are going to be located right in the middle of the lobule. The central veins will ultimately merge to form what we call the hepatic veins, as we can see right over here from this image with the liver. And the hepatic veins are the ones that empty into the inferior vena cava, which will then travel up to the heart, then to the lungs, and back into the systemic circuit. But now it's okay because this blood is a clean blood as the liver has filtered it. And your clue to figuring out if you're seeing a branch of the hepatic portal vein or a hepatic vein is to look for the inferior vena cava. Remember that the hepatic veins are the ones that empty into the inferior vena cava, not the hepatic portal vein, as this is the one that is bringing blood from the digestive system filled with toxins. Notice how the hepatocytes in a liver lobule will form a series of these irregular plates that are arranged like these spokes on a wheel. As you can see, these plates are only 1 cell thick, and in addition to typical endothelial cells, the sinusoidal lining includes a large number of what we call Kupffer cells, also known as stellate reticuloendothelial cells. And these are phagocytic cells that are part of the monocyte macrophage system, and they will engulf pathogens, cell debris, and damaged blood cells. Kupffer cells also engulf and retain any heavy metals such as tin or mercury, which are going to be absorbed by the digestive tract. Isn't this cool? It's literally impressive how our body thinks about every little detail. As we have seen from the previous slide, we will understand how the liver will have this metabolic regulation function by actually being the clearinghouse of the body. So circulating levels of carbohydrates, lipids, and amino acids are going to be regulated by the liver. We have also seen how all the blood leaving the absorptive surfaces of the digestive tract will enter the hepatic portal system and flows into the liver. And this arrangement gives the liver cells the opportunity to extract absorbed nutrients or toxins from the blood before it reaches the systemic circulation through the hepatic veins. Also, liver cells, which as we know are called hepatocytes, will monitor the circulating levels of metabolites and adjust them as necessary. In addition, we have that excess nutrients are going to be removed or stored, And mobilizing stored reserves will be able to correct any deficiency. Now, circulating toxins and metabolic waste products are also going to be removed for subsequent inactivation or storage or even excretion from the body. And lastly, we have that fat-soluble vitamins such as vitamin A, D, K, and E are going to be absorbed and also stored in the liver. When we were talking about the cardiovascular system and the blood distribution, we did mention that the liver was the largest blood reservoir in the body, and it actually receives about 25% of the cardiac output. And as the blood passes through the liver sinusoids, the phagocytic cells, which are the Kupffer cells in the liver, they will remove the old or damaged red blood cells as well as the cellular debris that are left behind, in addition to pathogens that are left in the circulation. In addition, the liver cells, which are the hepatocytes, they will be important to synthesize plasma proteins that will contribute to the osmotic concentration of the blood. They will also be important for transport of nutrients and for establishing the clotting and the complement systems that are important for preventing blood loss. Now, bile is going to be synthesized by the liver cells, so the hepatocytes, but this bile will actually be stored in the gallbladder, and it will be excreted into the lumen of the duodenum when you eat fat. So it's a big misconception about the gallbladder. It does not synthesize bile, it only stores bile. Bile will consist mostly of water with minor amounts of ions and bilirubin and an assortment of lipids that are collectively known as bile salts. Now, the water and the ions, they will come together to assist in the dilution and the buffering of acids that are coming in the chyme that enters the small intestine from the stomach. The bile salts that are associated with the lipids in the chyme, they make it possible for the enzymes to break down those lipids into fatty acids that are going to be suitable for absorption. Remember that if fats are too large, then what happens to them? They are going to be picked up by the lacteals and then will end up close to the heart once they enter the blood circulation. And bilirubin, we have already talked about it in the blood lecture, module 2. But it is a yellowish pigment that is made during the breakdown of red blood cells. It passes through the liver and it's going to be eventually excreted out of the body together with the bile, as it is a component of the bile. And if a patient presents higher than usual levels of bilirubin, then this may indicate different types of liver or bile duct problems because they're not able to excrete the bilirubin from the body. Moving on to the gallbladder, it has two major functions, which are bile storage, which we already talked about, and bile modification. So when the hepatopancreatic sphincter, which is also called the duodenal papilla, is closed, the bile enters into the cystic duct. So the cystic duct is what will carry the bile into the gallbladder. And when filled to capacity, the gallbladder will contain about 40 to 70 mLs of bile. And as the bile remains in the gallbladder, its composition will gradually change. So water is going to be absorbed from the bile, and the bile salts and other components of bile, they become increasingly concentrated. And this is what can lead to a gallstone. So this image actually shows a gallstone. And if you have a lot of gallstones produced in your gallbladder, you can actually have to remove the gallbladder. And it's okay if you have to remove your gallbladder. The only thing that you have to do is eat less fat, because bile is used to help with the digestion of fat. So if you just eat less fat, then you have no problems with the removal of the gallbladder. This just means that when you need bile, it will come directly from the liver. Now, bioejections occurs under the stimulation of the hormone cholecystokinin, or CCK, which we have already talked about, and it's going to be released into the bloodstream of the duodenum when the chyme arrives, and it contains large amounts of lipids and partially digested proteins. So this CCK will cause the relaxation of the hepatopancreatic sphincter and contraction of the gallbladder, and this releases the bile into the duodenum to help with the digestion of especially lipids. As we go over some of the lab models, there are some things that I want to point out. So this duct that's coming from the liver, this will be called the common bile duct. The one that's taking bile into the gallbladder is going to be called the cystic duct. And the one that's bringing bile out of the gallbladder into the duodenum is going to be called the common bile duct. So again, we have the common hepatic duct right over here, the cystic duct, and the common bile duct. And from here, we can see the common bile duct coming down over here, emptying into the duodenum. On this posterior view, we can see it as well, common bile duct emptying into the duodenum. On this model over here, this would be your gallbladder. So these would be your hepatic ducts over here. This is your cystic duct, and the one that's coming down over here would be your common bile duct. With regards to these other structures over here, we can see that this big blood vessel would be your inferior vena cava. Notice how this other blue vessel right over here is not merging onto the inferior vena cava. Therefore, this blue vessel over here would be your hepatic portal vein because it's not emptying into the inferior vena cava. And parallel to it, we have the hepatic artery proper. Therefore, always try to think about concepts, and then it makes it easier for you to understand the models. Lastly, we have the pancreas that will lie posterior to the stomach, as we can see from this image, extending laterally from the duodenum towards the spleen. And the pancreas is primarily an exocrine organ producing digestive enzymes and buffers, although it does serve an endocrine function, which we did talk about in Module 1. Yeah, I know, it does seem like centuries ago. Now, the pancreas has a large main pancreatic duct, and about 30% of us have an accessory pancreatic duct, which is located right over here. And both ducts will have their own sphincter that will empty into the duodenum, as we can see from this image. Now, the main pancreatic duct will empty its contents through the duodenopapilla, and the accessory pancreatic duct will empty through the accessory duodenopapilla. The pancreatic acini, which are the group of cells, they will produce a secretion that's called the pancreatic juice, which is going to be a mixture of water, ions, and pancreatic digestive enzymes. And these enzymes are going to be released into the duodenum through the ducts that we just talked about, where they will break down ingested material into small molecules that are going to be suitable for absorption. The pancreatic ducts, they also secrete buffers, primarily sodium bicarbonate, which are going to be important in neutralizing the acid in the chyme that's coming from the stomach and stabilizing the pH of the intestinal contents. Now, these pancreatic enzymes are going to be classified according to their intended targets, and it doesn't get easier than this. So lipases will digest lipids, carbohydrates such as pancreatic amylase digest sugars and starches, nucleases, they attack nucleic acids, proteolytic enzymes, they break proteins apart, And these proteolytic enzymes include, for example, proteinases and peptidases. These proteinases, they break apart large protein complexes, whereas the peptidases, they break small peptide chains into individual amino acids. Now, secretion of pancreatic juice, it will occur primarily in response to hormonal instruction from the duodenum. When acid chyme arrives in the small intestine, secretin is going to be released, and this hormone will trigger the production of watery pancreatic juice containing buffers, especially sodium bicarbonate, to neutralize the duodenum. Another duodenal hormone is CCK, which is going to be triggered by the presence of food or chyme in the stomach in the duodenum, and it will stimulate the production and secretion of the pancreatic juices to be able to digest the chyme that's entering the small intestine.